Let's talk about Python for bioinformatics application development. So, in this lecture, we are going to take you in the in-depth exploration of Python programming for developing some powerful bioinformatics application. We will cover some advanced Python concept, dive into essential libraries, and guide you through the robust real-world bioinformatics tools. So first of all, there are some advanced concepts that are now being implemented into Python applications. So we have list comprehensions. So we have to streamline the data transformation with concise, readable list comprehensions. Then we have some generators that harness the memory efficient power of generator function for large datasets. So these are basically some of those concepts that we are going to use in this course as well. There are a lot of concepts, we cannot cover all of those, but here are some of the main concepts that are also going to be used in the course as well. So we have generator that functions as the efficient power memory for our large data sets. Then we have decorators that enhance the functions to add functionality without modifying the core logic of our code. Then we have context managers that simplify the resource management for cleaner and more readable code. So all the code that I will be giving to you will be a plain, simple and clean code. You can add your own data, you can add your own analysis in that as well. Then we have some basic Python libraries that we are going to use throughout the course as well. So first of all, we have BioPython, that comprehensive library for sequence analysis, sequence alignment, sequence comparison, and biological data manipulation. Then we have Pandas. Pandas is a very powerful data structure and data analysis library that is used for efficient data handling of biological data. Then we have NumPy, number py, that's it is also called numpy, and the fundamental package for scientific computing that provides support for large multi-dimensional array and matrices. Then we have Tkinter. So standard Python library for creating graphical user interfaces. It provides us a set of tool and widgets for building desktop applications. Then we have developing some bioinformatics application. First we have design. So we have to plan our application architect and control flow for the optimal performance of that application. Then we have implementation. So we have to leverage Python advanced feature and libraries to implement our bioinformatics application. Then we move on to the testing phase. There we ensure the readability, robustness of our application through comprehensive testing. We test it again and again, and it gives us a better output. So we have to integrate some external tools and databases as well. So first of all, we have NCBI database. So we will use some query and retrieving data from NCBI extensive collection using Python, using BioPython library. Then we have to use the BLAST for sequence alignment from our Python application. And then we can move to the data analysis. We will praise and analyze data from external resources to extract insights and also enhance our application by using some of their bioinformatics workflows. So there is no worries about that. All the tools that we are going to make are going to be open source, and also all the libraries that we are going to use are also open source. So there is no need to pay for any tool. There is no need to pay for any extension that we are going to use. So recently I made a software that was a case study for the genomic variant analysis. So first of all, we will develop a robust pipeline. In this course, we are also going to do that. We will develop a robust pipeline to identify genetic variants from next generation sequencing data, that we will have our VCF files. If you don't know about VCF file and variant calling, you should check my course on variant calling, and you can get the idea from there. Then we have to annotate the variant from functional information and apply the filters to prioritize the most impactful changes. Then we will create a web-based application. We are not going to do that in this case, but it is a case study that where I did that, and we can analyze in a user-friendly way. But we are going to create an application that will tell us in our VCF file how many variants are available. For example, how many SNPs are there, how many indels, insertions and deletion mutations are there, and how many genomic variations are there. So then we have reporting. We have to report and draw meaningful insights from our data. So this course will have some hands-on experience. Some or not some, but all of them will be the hands-on exercises. We will implement common sequence manipulation and analysis tasks using BioPython. We will create a stunning data visualization library, like use some stunning data visualization libraries like MATLAB and Seaborn. Then we will query and retrieve the data from bioinformatics databases, and then we will build a complete bioinformatics application from scratch, following some of the best practices in the market. So, about your questions, we have some challenges in this field right now. We can discuss them on and by using the Python libraries. We will discuss them in the course as well throughout the course. So we will also explore some of the best practices. You can follow them to write a clean, maintainable, and efficient Python code. You can also get the guide from other. If you're stuck on anything, you can ask me the question. I will try to answer it. Then we have some career opportunities for a bioinformatics application developer. We can use Python in all the uh, analysis that we do. So we can leverage Python to conduct cutting edge research like genomics, proteomics, and system biology. We can be a bioinformatics developer, design and implement bioinformatics advanced tools and application using Python's capability. And then we have also bioinformatics analysts that utilize the Python data manipulation and visualization features to extract insight from complex biological data. So, about concluding the lecture for Python, we can master the Python programming for bioinformatics. We will unlock a world of possibilities from taking the data to analyzing it and making a exe file that we just have to click it, open it, and then run our analysis. Python is very flexible and it has a very powerful libraries that make it a indispensable tool for any aspiring bioinformatician. So embrace this knowledge and let's embark on a journey for a transformative bioinformatics solutions. So I hope you will enjoy this course. If you do now, please let me know in the reviews, and I will be waiting for you guys to make changes to the code and develop your own applications. What I am doing is just creating some of the sample bioinformatics tool. You can create your own and let me know how much changes that you did. My inbox is always open for you guys, and we will see you in the next lecture in the next video.